Hi everyone, in this video we're going to go over the viral life cycle and we're going to see how the lytic and lysogenic phases are both related to one another. So let's just get started. Here where they both kind of converge, we're going to see that we have a virus that comes along that attaches to the host cell. This virus is then going to inject its viral genetic material, whether it be DNA and RNA, that's what this is, and that will eventually find its way to the host DNA. So this one specifically, we're going to use circular DNA, and our example here is going to depict a bacteriophage. So once the viral DNA is injected into the cell, that will incorporate itself into the bacterial DNA. And at a certain point, it'll become a prophage, also known as a provirus. And what this means is that the virus is dormant, that the virus does not at this current time want to destroy the cell, but it wants to incorporate itself into the cell and as many cells as possible, so that once it finally does produce viruses, full viruses such as this, it can infect as many cells as possible. If it doesn't do that, then the host immune system will detect the virus and destroy it immediately. And as this is going on, the viral DNA will continue to replicate with the bacterial DNA. And in order to achieve this goal, the virus will have to incorporate itself into the bacterial genome. Once it does that, every time the bacteria divides, so does the virus. So I hope this comes out clear for you guys. We're going to use purple to show the mix of the red and the blue. And every time that happens, the virus is able to replicate. So the virus itself does not replicate on its own, but it is dependent on the bacteria to divide. So when the bacteria divides, that's when the virus divides. If the bacteria does not divide, then the virus itself does not divide. So here I'm just depicting some binary fission. 1 becomes 2, 2 becomes 4, 4 becomes 8, so on and so forth. So as the bacteria divide, so does the virus. And at this point, the virus needs to decide whether it wants to divide more or whether it has infected enough host cell so that it can cause an infection or that it can survive. And at this point, we still have it where the viral DNA and the bacterial DNA are still together. But if at this point, the prophage has decided to excise itself from the bacteria, it has also then decided to enter in the lytic phase of its life cycle. So from this point, if it says we've infected enough hosts, that we can produce enough virus, that we have enough to sustain life, then instead of dividing into more cells, it will then enter into the lytic phase, which at this point, since it has already incorporated itself into the host machinery, it will simply take over. It will do transcription, it will do translation, and all of that is an attempt to create the virus. At this point, we actually have assembled viruses inside of our bacteria. They have taken over the host cell machinery, and at a point where they have become too much for the cell to handle, they will eventually burst out from the host cell and enter the surrounding environment. From there, it can continue to infect more bacteria, which will again replicate the whole process over again. So, just to recap everything, the lytic and lysogenic phase are kind of one and the same, where one relies on the other, such that when the virus goes through the lysogenic phase, where it's trying to incorporate its viral DNA or RNA into as much bacteria as it can. And if it's at a point where it says that it still needs to continue to infect more bacteria, then it will simply wait and it will just divide more as the bacteria itself divides. But if at a certain point it has said we've infected enough in order for us to sustain life, it will then enter the lytic phase where it will take over the host cell machinery, assemble all the parts of the virus for itself. Once it has accumulated enough, it will eventually burst the bacteria. These free-floating viruses will then infect another bacterial cell and continue the whole cycle over again. Now, a good thing that you'll want to know is that the virus, it doesn't necessarily want to kill the entire organism. This actual cell is killed, but it's hoping that the whole organism itself stays intact. Because if it does, then it is also effectively killing itself. So there has to be a good balance between virus and host cell. Without that good balance between virus and host cell, the virus would not be able to replicate, either one, because it has killed off the host, or simply because the host has detected it and has eliminated it. A good example of this would be HIV, where HIV waits in the lysogenic phase for the longest amount of time, waiting for the appropriate cells to accumulate. Because if those cells don't accumulate, the virus says, nope, we don't want to infect yet because the host immune system will kill us if we do. So we're going to lay dormant. And we're going to lay dormant until the proper conditions arise. Now, if you guys want, the next video is going to come out and it's going to say what those proper conditions are. But if you want, feel free to shoot me an email getting back to me saying, hey, these are the conditions that are important for HIV in order for it to enter in the lytic cycle from the lysogenic cycle. So I hope this video was helpful, you guys. If you have any questions, please feel free to shoot me an email and I'll get back to you as soon as I can.